Hello everyone. In this video, we cover the highlights of Chapter 3 on nucleic acid structure and function. This covers sections 3.1 to 3.33 in Essential Biochemistry. So starting out with nucleotides. So the reason this is important to study is because nucleotides are involved in every facet of cellular life. So we see them in redox reactions, energy transfer, intracellular signaling, biosynthetic reactions, structural and catalytic roles. And there'll be lots of examples of those in the class in the days and weeks to come. And of course, we use the nucleotides to make nucleic acids, and that's involved in storage and decoding of genetic information. And it has structural and catalytic roles as well, which we're still appreciating, I think. So starting with the building blocks, the term nucleoside consists of a nitrogen base and a sugar, whereas a nucleotide is a nitrogen base, a sugar, and 1 to 3 phosphates. So here's examples of purines and pyrimidines. So the purines are these two-ring structures, and the examples are adenine and guanine, whereas the pyrimidines are a single cyclid structure, and the examples are cytosine, thymine, and uracil. So when we take these bases and we attach them to sugars, if we're looking specifically at the purines, and this is the numbering system right here, now nitrogen number 9 is where we attach the sugar, so where this arrow is. So we'll form a covalent bond with either adenine or guanine at the sugar at nitrogen number 9. For the pyrimidines, and again, this is the numbering system right here, this is nitrogen number 1. So for nitrogen number 1 for cytosine, uracil, and thymine, that's where we form the covalent bond with the sugar. So here's a nice table, I think, of all of the bases and how we build them. So these are the bases, and we have adenine, guanine, cytosine, uracil, and thymine, and these are the abbreviations. And when we attach, so this X right here could be ribose, or maybe deoxyribose, depending on what nucleic acid we're going to build. And the X shows how we covalently attach either the ribose or the deoxyribose. And remember, this is a nucleoside, so we'll change adenine to adenosine, guanine to guanosine, cytosine to cytidine. This is a little confusing. Uracil to uridine, and thymine to deoxythymidine, because typically we have a deoxyribose there. If we're going to then add to our X-ribose 1 or more phosphates, then we have a nucleotide. So this just shows if we have monophosphates. So we have adenosine monophosphate, or AMP, guanosine monophosphate, or GMP, cytidine monophosphate, or CMP, uridine monophosphate, or UMP, and deoxythymidine monophosphate, or DTMP. We can, of course, add more than one, in which case we use di or tri to indicate the number of phosphates. So we refer to nucleosides as ribonucleosides if ribose is the sugar. So this is what the ribose looks like. And I showed you the numbering system for the purines and the pyrimidines. So we have to number the ribose too, but we want to make sure that we don't get our numbers confused. So that's why we use prime. So this is 1 prime, 2 prime, 3 prime, 4 prime, and then this is 5 prime outside of the ring. So if you just have a plain number, those are atoms in the nitrogen bases, either the purines or the pyrimidines, whereas the prime refers to the sugar. So this is the sugar for the ribose, and the bases that will bind to ribose will be A, G, C, and U. So this is our ribonucleoside. So this is adenine right here, and that's our nitrogen number 9, and we have a covalent bond to our ribose. And again, ribose is going to have, on this 2'd carbon, will have a hydroxyl right here. So this is adenosine. It's a ribonucleoside because it consists of a nitrogen base and a sugar. A ribonucleotide will take our base and our sugar, and at the 5' carbon, we'll do a condensation reaction and we can attach a phosphate. So this is a ribonucleotide. So again, we have a sugar, a nitrogen base, and anywhere from 1 to 3 phosphates. So this is a ribonucleotide, or AMP. So we have adenine, ribose, and then we have 1 phosphate. Now those phosphates, it doesn't show in that last picture, but the phosphates are typically ionized at physiological pH. This is another ribonucleotide, but we have 2 phosphates. And so this is adenosine diphosphate. And the bond between this is called a phosphoanhydride bond. And if we have 3 phosphates, we have a ribonucleotide ATP adenosine triphosphates, and we'll have 2 phosphoanhydride bonds. So we have 2 phosphoanhydride bonds between the phosphate groups. If we have a deoxyribonucleoside, that means instead of ribose, we have deoxyribose. So a ribose, remember, would have had an OH right here. So deoxy means that we don't have that oxygen. So we just have a hydrogen. So this is our deoxyribose. If we covalently attach it to a base, we have a deoxyribonucleoside, remember, which is a ribose plus a base. So we typically have A, G's, C's, or T's as examples of deoxyribonucleosides. So here is a deoxyribonucleoside, deoxyadenosine. So here's adenine, and this is our sugar, which is deoxyribose. Again, so we don't have an OH right here. We just have hydrogens. So our naming system, here are our nitrogen bases, and then we would call these, if we have a sugar plus a nitrogen base, we have a deoxyribonucleoside. So we can have either deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. A deoxyribonucleotide means that we have a phosphate, and that oxygen actually would be an O- at physiological pH. But this shows an adenine, a deoxyribose, and 1 phosphate. So here's our table again, in which we show that we could have X as our ribose. With thymine, we typically have deoxyribose. But we could have deoxyribose for adenine, guanine, and cytosine as well. So the functions of nucleotides just by themselves. So ATP is adenosine triphosphate. So here's our adenine and our ribose and our 3 phosphates. And the hydrolysis of these phosphoanhydride bonds releases free energy, and that can be captured to do cellular work. So we'll see these reactions later on in the class. But ATP can be hydrolyzed to ADP. So if we hydrolyze the one of these phosphoanhydride bonds, we release an inorganic phosphate. So we're just abbreviating inorganic phosphate. But that's its chemical structure down here in red. Or we could hydrolyze this phosphoanhydride bond right here, in which case we would release, our products would be AMP and a pyrophosphate. So those are just nucleotides that have their own cellular roles. We can use nucleotides as building blocks for nucleic acids, and we can make DNA and RNA. And here, when we link our nucleotides or our deoxyribonucleotides, those linkages are phosphodiester bonds. So here is the chemical structure of a nucleic acid. So here you can see that the sugar is ribose, and the way that this works is we have a 5' phosphate on our on this end right here. The ribose would have had a 3' hydroxyl, but we can do a condensation reaction with the next phosphate on the next nucleotide. So what we have is we have 5' 3' phosphodiester bonds, and this is in the 5' to 3' direction. So our 5' end is going to have a phosphate, and our 3' end is going to have a hydroxyl. So this would be a stretch of RNA, because we have uracil and we have ribose as our sugar. So again, that's our 5' end, which will have a phosphate, and the 3' end will have a hydroxyl. If we look at DNA, we can look at some of the history. So in the 1940s, before people knew what the structure of DNA looked like, Chargaff established that if you took the amount of adenine plus guanine, it was equal to the amount of thymine plus cytosine. So we call this Chargaff's rules. We know now, because of DNA's double-stranded nature and the complementary base pairing, but at the time, they had no idea. And it was kind of interesting because the base composition varies, but Chargaff's rules always held true. So the double helix of DNA was determined by Watson and Crick in 1953, and they used X-ray crystallography data taken by Rosalind Franklin, who was working at King's College London with Maurice Wilkins at the time. So if we look at a very stylized view, we can see that A pairs with T and G pairs with C. And what we have is two polynucleotide chains that wind around a central axis to form a double helix. And the strands are anti-parallel. That means you'll go 5' to 3' in this direction and 5' to 3' in that direction. And we form our complementary base pairs where A pairs with T and G pairs with C. So here's just some pictures from history. So here's Chargaff. So this is Rosalind Franklin, and this is the very famous photograph that she took. And if you're skilled at reading X-ray crystallography, which I am not, but I've actually read how you would interpret these values right here. So this picture is very, very expertly taken, and it clearly leads to the structure of the double helix that we see here from the first model that Watson and Crick built after they were able to view this photograph. The other thing that was important was that at the time there was some disagreement about what tautomers the base structures had. There's ketoenol tautomerism, and Jerry Donahue, who actually happened to be working in the same lab as Watson and Crick at the time, were able to provide the correct tautomeric structures that give us the hydrogen bonding pattern that we know today. So when we take our two nucleotide strands and pair them up and form our double helix, we see we have a major groove and a minor groove. The major groove is a good place for proteins to bind because proteins are very important in both in maintaining the structure of chromatin and the regulatory features of DNA. The helix winds in the right-handed direction, so that means if you take your right hand and point your thumb up, that you'll the helix will turn in that direction. The other way to look at it is imagine that you have a pole down here with this helix, and if you did have that, then the helix would cross to the right-handed direction as it crossed in front of the pole, which means it's right-handed. So this is down here, it has the detailed chemistry of the complementary strands of DNA. So again, we go from the 5' phosphate to the 3' hydroxyl in this direction. Here we go 5' to 3' in this direction. So we have our anti-parallel complementary strands, and this shows the detail of the structure of the deoxyribose and the bases. And we can see we have 2 hydrogen bonds between A and T and 3 hydrogen bonds between C and G. Now, there are important forces in stabilizing the double helix, but the hydrophobic effect is very important. The bases are located on the inside, and the charged sugar phosphates are on the outside. But the most important are these stacking interactions. So stacking interactions have been physically measured in the lab, and stacking interactions are much stronger for GC base pairs. The hydrogen bonds are less important. We do know that there are 2 for AT and 3 for GC. This is often given as the important stabilizing force. But if you separated the helix, those hydrogen bonds could just as easily form with water, so they're less important. It's the stacking interactions that are most important. And there are electrostatic interactions that are important too, between negatively charged phosphates and positively charged ions or proteins that have positively charged domains. We can look at the stability of DNA by taking a sample of it and melting it. And what happens is that if you have a double-stranded DNA, those stacking interactions unwind, and it turns out that that the absorbance at 260 is actually higher for single-stranded DNA than it is for double-stranded DNA. So you can measure whether you have double-stranded or single-stranded DNA by looking at the change in absorbance. And if you look at the inflection point of this curve, you can figure out the melting temperature. So this would be when the DNA is half melted. So this particular sample of DNA would have a melting temperature a little higher than 70 degrees Celsius. So the higher the TM, the greater the stability of the DNA. So if we look at lots of different organisms, we can see that the GC content varies quite a lot, but we do see a correlation between the GC content and the melting temperature. And again, it's because you have, if you have a higher percentage of GC base pairs, the stacking interactions for GC base pairs are much stronger for GC than they are for AT, and that explains the stability of the DNA molecules that have the higher melting temperature. But of course, Chargaff's rules still apply. G pairs with C and A pairs with T. You can renature DNA if you heat up the DNA, and if you slowly cool it down, the complementary strands will find each other, and you will be able to renature it. But this won't happen if you rapidly cool it too much. You'll get some improper base pairing. So you have to be patient and cool it slowly. But if that happens, you can just rewarm it and try again. So moving on to RNA, RNA occurs primarily as single strands, but it does assume a secondary and tertiary structure. So we do have base pairing, but that's intramolecular, and that gives us our stem loop structures. So it is a polymer of ribonucleotides, and interestingly, RNA has been shown to have some catalytic properties similar to enzymes, to protein enzymes. So here's our single-stranded RNA. So we have A, C, G, and U, and here is our base, which is ribose, and this is going again in the 5' to 3' direction. And we can take this and we can form a stem loop structure. So we can base pair some regions of the RNA, but again, this forms in a single loop. So our DNA carries genetic information. Up until the 1940s, it was assumed that genes were made of protein because protein had maybe greater diversity with 20 amino acids, whereas nucleic acids were thought to have monotonous sequences, only 4 bases, and unlikely candidates for transmitting genetic information. But there's some experiments that you can go back and look at. Griffith's transformation experiment and the identification of the transformation factor as DNA, that challenged that dogma in the 1940s. But it still wasn't understood until the structure of DNA became known. So the importance of DNA is that it serves as the depository for all of the cell's genetic information. So we refer to the genome as the cell's DNA content. So when DNA makes a copy of itself, as it does during mitosis, the complementary nature of the DNA lends itself very well to making a copy of itself. So if we separate the strands and then make new, which is shown here in red, we can use the old DNA as a template, and we can make an exact copy, again with A pairing with T and G pairing with C. So we refer to this as semi-conservative because the 2 copies of the new DNA have one old strand and one new. So this gives rise to the central dogma. So DNA makes a copy of itself, as I just showed you, but we can use a strand of DNA as a template to make RNA, and for messenger RNA, we can use that as a template to translate that into protein. So so DNA makes RNA and it makes proteins. Using DNA as a template refers to transcription, and then RNA, especially messenger RNA, is translated into protein. So I have some pictures here to show you. So, these have some names that are maybe a little bit confusing. So, if we look at the DNA right here, again we can see 5' to 3' in this direction, 5' to 3' in this direction, and during transcription, we would have used only one strand. So we would have used this bottom strand, because if you look at the messenger RNA right here, you can see that we must have used this bottom strand, which we call either the non-coding or the template strand. We must have used this bottom strand because we get a messenger RNA that's complementary to it. So you notice that the coding strand looks exactly like the messenger RNA. If you look at 5' to 3', it's the same direction, and the sequence is the same, except for that we replace the T's with U's. So it's convenient that the coding strand and the messenger RNA have the same sequence. Then we count off by codons. So we have codons on the messenger RNA and complementary anticodons on the transfer RNA. So the transfer RNAs will bring in the amino acids, and then we will form peptide bonds between them to get our protein. And here's another example right here. So this shows that we're using one strand of the DNA as a template to make the RNA, which is shown here in red. So again, the bottom strand is the non-coding or the template strand. That's what we use. The coding strand or the non-template strand is the strand that we don't use. But again, you can see that the sequence of the the non-template or the coding strand is the same as the RNA, except for the RNA has U instead of T. Here's another example right here, where if we look at the nucleic acid, we consider that the codons, this sequence of 3 bases are our codon, and the transfer RNA has the anticodon, and that carries the amino acids, which are linked together via peptide bonds to make the protein. So we can call this translation because the codons don't directly recognize the amino acids. We need the anticodons to bind to the codons, and we need the transfer RNA to carry the correct amino acid as specified by that anticodon. So here is our standard genetic code. So if we have 3 bases, we have 64 different possibilities of how those could be arranged. And we only have 20 amino acids, so we have a code that's redundant, where you can have more than one codon would specify a single amino acid. And of course, we have some stop codons as well. So when this occurs, if this is our messenger RNA, the messenger RNA is sort of clamped between the small and the large subunits of the ribosome. And these are our codons right here, bound to the anticodons on the transfer RNA, and the transfer RNA brings in the amino acid. So this is sort of a work in progress, where this amino acid will be added to this growing polypeptide chain, and then we'll just move this messenger RNA over. This transfer RNA is ejected, and the next one comes in, where its anticodon base pairs with the codon on the RNA. So I wanted to tell you a little bit about RNAi, or RNA interference. This is a way of silencing genes. So we have small interfering RNAs, and these are regulatory RNAs. They're snipped from a longer double-stranded RNA. So we actually do have some double-stranded RNA. An enzyme is called dicer, and then the antisense strand is used by an RNA-induced silencing complex called RISC, and that guides messenger RNA cleavage. And if you cleave the messenger RNA, then you can't use it for translation. And not only that, when you just have pieces of messenger RNA, the messenger RNA is degraded. So this is a way to halt translation in its tracks, to prevent the messenger RNA from being able to be used as a template for translation. So this is naturally used by the cell in regulatory processes, but it's currently being investigated so we could use these RNA molecules as drugs to inhibit protein synthesis in the treatment of certain diseases. So I'm going to show you this video. And I'm starting it at the 2-minute mark.
But this is also where some small RNA molecules can have their silencing effects.
So this is the ribosome right here.
There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC matching allows microRNAs to target hundreds of endogenous messenger RNAs.
So I decided to stop the video there because it goes on to talk about other types of RNA regulation. But I think you can see in the video how you can use RNA interference for gene regulation, but you can also manipulate this in terms of using drugs to inhibit protein synthesis. So here's another picture right here. So this shows the gene silencing. So we have our double-stranded RNA that was shown in the video, and then we have this cleavage into these small interfering RNAs that bind to the Argonaute protein that was shown in the video, and then you strip off one of these strands. So here, this shows the red strand. So if you have the red strand and we have the bases are complementary, then that small RNA will bind to the messenger RNA, and then when you bind to the RISC complex, then the messenger RNA is cleaved into small pieces. So we can't translate this, and small pieces of messenger RNA are degraded. So that's how we inhibit translation. So I hope that this is helpful, and I will see you in class.